Okay, hello everyone. It's 3:30 Eastern Time. Time to get started up again. People are going to continue to trickle in. I imagine if they're like me, they just have so much fun over in Gathertown, they find it hard to pull themselves away. I don't know if you picked up on the sarcasm in that. So in this session 5, we have a fantastic speaker in store for you. John is going to walk you through a complete example of PhysiCell model interactively. So I anticipate a wonderful show. I know he's been working hard on this, and I will leave it up to John to decide how he wants to do the Q&A, on the spur of the moment or hold them up. But anyway, he can decide that. And we're up to about 28. So as I say, 29. So John, whenever you feel comfortable, let you play.
Thanks. Thanks for that great introduction. For Khan, are you going to be recording this session again, or shall I just record it?
I started actually.
Okay, I didn't hear the tone, and I don't see. Maybe it's because I'm sharing my screen. I don't see.
Yeah, it's definitely blinking. It's recording.
Okay, great. Thanks for reassuring me. Great. Oh, that's interesting. Let's see. I tested my setup before, so please pardon me while oh, great. Okay. I have some notes down here and they disappeared temporarily. Anyway, again, Randy, thanks for that great introduction. As he said, also welcome to session 5 of the 2021 PhysiCell workshop. I'm John Metzcar. I'm a graduate student at Indiana University in the Math Cancer Lab. So I'm one of Paul's students. It's a great team. So today I'll be going over a complete PhysiCell example in an interactive format. In terms of questions, I like the way that it was set up before, where questions could accumulate and in the chat. And I've built in definite breaks in the slides for anybody to ask questions. My hope is to bring everybody along so that hopefully we'll have enough time to have everybody come through the entire session together. And if as needed, we'll we'll move on if we have to, but hopefully everything can be done live. And I've got this. Oh, great. So the goals of this session are to we're going to review the intermediate modeling workflow that Paul went over briefly at the end of his session this morning. And then we're going to basically do that intermediate modeling workflow. So we're going to build a model from start to finish. Of course, as with any problem, you need to start by stating the problem. So start by stating the problem in words, and we'll plan the model or otherwise what we're attempting to model mathematically and computationally. We'll adapt the template project with the model builder, the graphical user interface that Randy and colleagues developed over the spring, and then run and view our results. And then time permitting, we'll explore the model a little bit in a couple different scenarios, use the model builder to change the domain and cell placement, and contrast non-stochastic and stochastic proliferation rates. So in terms for reviewing the intermediate modeling workflow, so this is really, as stated, it's really suitable for creating a new PhysiCell model without getting into the C++. But the limit is that there are no changes in cell behaviors in response to the environment or cell interactions. So basically, cells have a static phenotype. So as stated in the previous slide, this work in general, all modeling workflows are going to start with planning the model. So building it conceptually and then translating that concept into mathematical rules or quantitative numbers that can then be implemented in our computational framework. And in this case, of course, that's PhysiCell. And the way that we're going to interact with PhysiCell is through the model builder GUI, the graphical user interface. And so we'll go through editing the domain or setting the domain, setting the microenvironmental elements, the substrates that are available, and then make an edit, starting with the existing cell definitions, edit them, and then make new ones. Then we'll be able to run, that'll be outside of the model builder, that'll be at the command line, and then view and evaluate the results. So for our model, let's use this concept of carcinogenesis, what I'm calling the steps in carcinogenesis model. So diseased cells and tissues develop from healthy tissue. So thinking about that, to be honest, this notes thing isn't working with me, so I'm gonna do my best to just maybe even ignore it. So diseased tissue starts from healthy tissue. So we can think about first a well cell, a cell that isn't diseased. So we'll call that non-cancerous cell the wild type cell. And this wild type cell, we're thinking about it coming from an epithelial cell line, or the kind of cells that tend to stick together and form layers and divide slowly. So that's exactly how I've encapsulated it here. So we have a non-motile, non-moving, slowly dividing adhesive cells for this cell line. And then we could think about cancers developing progressively from small changes over time in a particular cell line. So skin cancer might develop as there's damage to the skin, and then, but it doesn't happen all at once. And so we're going to attempt to recapitulate that here in PhysiCell today. So we're going to progressively enable small changes in behavior and form 3 aberrant cell types: a hyperplastic type that isn't cancerous per se, but has increased proliferation. Something's not quite right. Then a cancerous type that is less adhesive and motile, no longer forming a tissue layer. It's much faster, it's faster dividing, and also it suffers from an increased relative death rate because it may be lacking the checkpoints, the stopping that a typical cell would have. And then an aggressive cancer type that is resource seeking, resource driven, and very quickly dividing with a further increased relative death rate. And to support our cell models, we'll also employ a microenvironment. And if we're going to have resource-driven cells, we're going to need to have a resource for the cells to seek. So we're going to consider a surrogate for nutrient for cancers. In this case, we're going to use oxygen, and there are others that people have used and could use. And then for the domain, at this time, there's PhysiCell, as mentioned earlier, needs a Cartesian domain, so we'll use a Cartesian domain. But other than that, I mean, there aren't really any restrictions at this point in the model. And we aren't really talking about a particular shape of the tissue, which, if I noticed this before, would have actually, that's of course an initial condition. But there's no particular initial condition that's required right now as conceived. So now, next in our general modeling and intermediate PhysiCell modeling workflow is mathematical modeling, where we translate the conceptual model into a quantitative or rules-based model. Just to state it, of course, we all know that there's multiple layers of influence on cell behavior. As an example, gene mutations or changes in gene expression can change mechanistically what a cell or set of cells or even a whole tissue might do. But for this model, we're going to simplify to the level of what can be observed just by looking at the cells only. As Paul mentioned earlier, at its core, PhysiCell is by itself is a phenotypic driven modeling paradigm. So we want things like growth rates and cell speed that can be tracked to or set to an individual cell level. And then going further through the whole modeling workflow, by implementing this model, this mathematical model into a computational model, we'll be able to evaluate which behaviors of the simulated cells and cell populations match our expectations and which don't, and then iterate on that. So this is of course, the details of the cell types. So I'm going to list out the cell types, then the microenvironment, and then the domain. So this has 4 cell types. and it's the quantitative encapsulation of our biological model. And I've paired each description, each number with a description of how it fits in our biological model. And in particular, just I'm not going to go through all the details, but noting that in this case, the wild type has the longest cell cycle duration, then each one decreases, going to half of, for the aggressive cancer, half of what the wild type is. Note also, in the interest of time, I've selected very short cycle times, and so that we can get results quickly in this pedagogical setting. And the real world, the rates would be whatever was appropriate given modeling goals and available information. For a microenvironment, we're going to model our resource as oxygen. In this case, we're using the default values from PhysiCell. PhysiCell historically has used oxygen a lot. So this is the value that was used, I believe, in a 2012 paper and in the 2019 PLOS computational biology paper. And then our decay rate is for the diffusion coefficient for oxygen and tissue. And then our decay rate is going to be set by getting a length scale of 1,000 microns, which is typical for just tissue. For our initial boundary conditions, we don't need a particular initial condition. In this case, I'm just going to go with 0. We could do different ones. Decay would take care of it for us. What we really need is a gradient. So we'll set our boundary condition to be 38 for a reasonable value. And then finally, cells. There are many different cell configurations that would probably work. For right now, we just need a random, we're going to start with a random placement in the domain of 2 cells per type. In this case, because of stochastic birth and death, if we don't have 2, the cells might die before they replicate. So we need at least 2. And then our domain is going to be set to 2D, and we're going to have a relatively small domain of 500 microns by 500 microns. So at this point in time, are there any questions about the general intermediate workflow, or about the biological conceptual model, or the mathematical model that I presented? And I don't see anything in the chat, and no one's raising their hand, so I'm going to move on.
Actually, there was one question.
Oh, great, I see it.
Yep.
Thank you, Randy. Please just definitely keep me, that's right, Daniel. Right now, we're not, I'm not conceiving of it as actually mutating. Right now, that's not something that can be handled in the model builder at this time. So we're going to present the steps. Thanks for asking this. We're going to present the steps of carcinogenesis as if we're sampling from a population, or even as though we had already established cell lines and wanted to do like a phenotypic assay to see how they varied. So you can imagine in your favorite cancer biology lab at your institute, you want to see what the differences are, and so you do 4 different cell types and try to quantify that. Or in our case, we're going to actually qualitatively look at it. Is that so hopefully that addresses your question. Great question. So now we're going to move down into the sort of the meat of the PhysiCell. But first we have to get started. First, we need our toolkit. In order to build something, we need tools. And our toolkits in this case are PhysiCell and the PhysiCell model builder. So just to state it, we assume that you've already had your system set up. And also for some commands to run directly as written in the slides, the PhysiCell and PhysiCell model builder directories need to be at the same level within the file hierarchy. Otherwise, you'll have to modify them slightly. We're happy to help with that, of course. And then for anybody who ends up watching this later, looking at this later, all of our resources that are needed to run this tutorial are available here. So let's open up our toolkits and get started. So I'm going to attempt to switch to the same way that Paul was doing this. So bear with me while I'll continue my share. Great. I think, is everyone seeing slides on the right and then 2 terminals on the left?
Yep, looks good.
Thank you. So as Paul previously suggested, I suggest you open up 2 terminals or shells, whatever's appropriate for your OS. And one of them will build the PhysiCell, and then the other one we'll use for the model builder GUI. So if you have the slides open, my intention is that you should be able to, so first you navigate to your PhysiCell directory, which I conveniently already have open. Should be able to copy and paste directly from the slides, and then see output similar to what you see here.
Hey, John, if it's easy, could you increase the font size a bit?
Oh, that's a great idea. Would you, is that a very straightforward thing to do without getting into settings? I do not know how to do that. And I'm also,
Yeah, just try your command
okay.
plus.
Oh,
So that- yeah,
great.
the other one too. That's
this-
that's plenty large, I think.
Okay. Then this will
Screw up your nice symmetry, but
yeah, but it's much more important that people get what they need, obviously.
thank you. That's great.
Thank you. I'll take some time to readjust. Perfect. Great. So make reset will get you back to where PhysiCell was when you downloaded it. If you run execute make data cleanup, it will remove everything in your output folder. So don't run it if you want anything in that output folder. Rename the folder and then run the command. Make template will call up the template project and everything that's needed in order to make, and then make, which I'll just type in, will compile the project. Should have tried that -J4. I hadn't seen that before. But that didn't take very long, thankfully. Actually, I compiled it before. So then on our second window, navigate to PhysiCell config. And then this, if I can get it to not copy that asterisk, will load the model builder GUI. And note that in my case, I have in my model builder GUI, I have the version number in there. So if you don't, you might have to alter that. All right, now let's see if I can use, how do I advance? That'll work. Great. So we're ready to get started. So what I have going on here, let's see if I can blow this up a little bit. That helps maybe some. Is on the left side, I have instructions on what you need to do. And then on the right side, I have an image of what the final model cell builder should look like when you've completed all the instructions. The setup domain is a single slide. So I'm going to decrease this. I think that that'll be worse then. Also, I've tried to put all needed parameters on the side as well. So hopefully it's easy to follow. Please let me know if I need to adjust. So Xmin will be going to -250, Xmax to 250. And then this is square. So the settings are the same. As stated here, the config basics gives you kind of access to some meta parameters and as well as the computational domain. So I'm going to set the max time to 4,320. Oh, and I missed that I need to put on virtual walls. Also make sure that cells don't leave the domain. And that's that. That's all I need to change in that tab. Now, microenvironment. So go to microenvironment tab. Here's where you would add substrates, the diffusing chemicals that might be in an environment, and you get complete control over them at the level that PhysiCell allows. At least the general user. So I'm going to rename it. Setting up oxygen, renamed it to oxygen. And then because it's a default in PhysiCell, the default substrate, it already has the correct diffusion coefficient and decay rate. And all I need to do is change the boundary condition to be 38. And then everything else I'm going to leave the same. Everybody, I do see questions. Okay, Randy, is there anything that it'd be helpful for me to address or anyone else to address?
No, there's actually no questions,
Okay,
at least unless you're seeing something I'm not.
I'm gonna trust you on that for sure.
Yep.
Great, thanks. And if anyone isn't at this level, please raise your hand or otherwise indicate that we need to take a little pause. That's what I'll try to be doing as we go through the implementation so that everybody's able to stay on this. So barring reaction, always happy to go back, but barring reaction, I'm going to move on. So we'll set up the cells. And there'll be a pause after this set too, because this is arguably the most important. The setup cell types gives you access to the whole phenotype. We're not going to modify everything. Most of the defaults are acceptable, if not just even plain desired. But I'll be going through what we will be changing. Underlined is what each particular slide is on. So here we're going to rename the default cell to wild type. And then I'm going to click on cycle, which is already on. Live cells. And then here we get the mathematical, again, continuing down, now implementing the mathematical model. All right, let's move on to the death subtab. This value is in inverse units to the duration, but it actually ends up being 0.01% of the birth rate. And so all this is, instead of editing the XML like Paul did this morning, this allows you to just, in a much more human readable fashion, change things. So we're going to mess with cell adhesion a little bit to make these cells a little bit more adhesive. And for the purposes of visualization, I chose to keep them at the same distance apart, the same equilibrium distance. And I think that's it. So happy to take, most importantly, I want to make sure that everybody's following along, that everybody has this set. In particular, this first cell is where everything is going to get copied from. So if you don't have this ready, then other things might end up being harder. So first of all, is there anybody, please raise your hand or indicate if you need more time.
There have been a few say they're trying to catch up. Daniel asked, are millimoles the units here? And I'm not sure which.
Great. I can address. So in this, so I'm back on the microenvironment tab, and I think maybe it was on the slide. If it wasn't, I intended it for it to be. Millimoles are not the unit here. It's millimeters of mercury. And right now, so you could go in and edit the XML so that it reads correctly in your in standard out. But right now, the model builder isn't set up to change that at this level, but you can edit it in the XML. And I think you'll be seeing a demonstration of that tomorrow, I think. Great, what else?
I'm tracking down a link to the model builder to share.
Okay.
I think that was somewhere, but I'm not sure where.
Great, I'll start looking for questions then. And I can get that link too if it would be helpful. I think it is worth pointing out that this is a newer tool.
I got it, I'm sharing.
Okay, great, thank you.
You should grab the latest release there. Now this is so new, we do not have a tutorial specifically for it yet.
One of the reasons that I definitely wanted Randy to help out in my session was because he absolutely is he knows the most about this and is definitely the right person to have for help on the GUI. Thank you, Randy. Okay, and then what command to open this GUI? I can paste that into the chat. So Yep, that'll work. And then if you execute that command as written by Nikki, thank you, Nikki, for posting that. You have to be within the PhysiCell model builder directory. The other, oh, I sent that directly to Furkan. This command, if you are in the PhysiCell/config, will let you save the XML that the GUI generates directly into the PhysiCell where the PhysiCell executable can use it. If you load it from the PhysiCell model builder and load the GUI, then you'll end up needing to move the XML to PhysiCell/config in order for the executable to access it. All right, I'm going to wait another second to see if there's any couple seconds. I'm only on 13 of 47. There's plenty of time. Wait, no, I'm even further than that. All right. So I'm going to navigate. So I'm going to move on with the rest of our setup. So the rest of this will basically be really similar to what we did before to set up the wild type. It's just going to be like, as stated, kind of progressively building on different minor changes to the behavior, to the phenotype in order, and then we'll be able to see at the end what kind of how the cell line as a population ends up behaving differently than among the cell types. So oh, I'm in the wrong one. Wrong question. Great. So we're going to set up the hyperplastic cell type now. So I'm going to click copy while selecting wild type. So now I get a new cell type, and it has the same parameters as before. This one is hyperplastic. No one wants a hyperplastic cell. We don't have to set we don't have to set the cell cycle model again because it inherited it. And so all we need to do is alter the time, the duration. Move to the death tab. And this is just to keep our birth and death rates aligned percentage-wise. So there's no more death than the other cell line by doing this change. And then for fun, I put this in here, and so we don't need to change, just to point it out, we don't need to change anything here because it inherited directly. So now we're ready to set up our third cell type. Again, in this case, you want to make sure that I've written it such that you will be altering the hyperplastic type, continuing to build these, if you will, population level based steps in carcinogenesis. So copy the hyperplastic, double click in, rename it cancerous. I'm going to close that just in case. And now I'll go back to cycle, decrease the cycle duration again. This case is going to be a significant change as we increase the rate of death by an order of magnitude, percentage-wise. Since this is a cancerous cell, it's going to want to not stick as much. We're going to decrease the cell-cell adhesion a little bit. But the equilibrium distance of the potentials will PhysiCell will automatically make it work out so they have the cells have the same equilibrium distance. Oops, I'm getting ahead of myself. So mechanics, it should look like this. And then motility. We've got several things to change here since we haven't altered this yet. I'm going to set the speed to 0.2 microns per minute. Persistence time to 10. That's how many times PhysiCell calls the motility update function, or it gets called stochastically, so not how many times, but rather you could think of it as the frequency or time between calls. And then the migration by a small matter, because we're only enabling my motility. So now the cells will move randomly in essence, not move exactly randomly. All right, we're on our third and final cell type, or fourth and final cell type. So again, following the same format, we take cancerous and copy it. Rename. Alter cycle. Alter death rate. In this case, it's much higher. It's 4%. I've set it to 4%. Doesn't seem like 4%, but whatever it is, percentage-wise, is what we're going to use. And we don't need to change mechanics. We do need to change motility to allow chemotaxis. We're enable. I got ahead of myself. Let's set this to 0.25, which is I'd call it a noticeable amount of bias, but the cell will still be somewhat random. The cell motility will still be somewhat random. And in this case, we only have one substrate. You could enable chemotaxis on a per cell type basis. You can set how it chemotaxis within the model builder GUI. So different cell types can chemotax to different substrates. Great, and then I think I have questions next. Oh, I went ahead and finished it because there's one user parameter that needs set. So up at the top level in the hierarchy of tabs, go to user parameters, and we need to set this default value from 5 to 2 so that we don't have too many cells. And now setup is done. Yay! So Randy, so first of all, if you of course, if you like me to pause for longer, please raise your hand and we'll make sure that you can have the chance to get caught up. I forgot to mention, I did put in rescue. I should have mentioned this right away. I did put in rescue XML so that you can still run the simulations. And ideally, even as long as you're not having trouble running the model builder GUI, you could load that XML and hopefully you'll see everything that I've already put in. So that rescue XML is in the code section in the session 5 directory of the GitHub repository. So please use that as needed, and hopefully you're able to follow along live.
So John Bernardo is asking if it's possible to basically define differential adhesion in the model builder between two different cell types.
So great question. So by differential adhesion, what I believe you mean, this is this is what I'm intending to say when I say this, is that if you have cell type A and cell type B, that cell type A has a different adhesion parameters with cell type B than it does with itself, for an example. So assuming that's what you mean, the model builder does not allow that. And my understanding right now is that that's not an inbuilt function in PhysiCell. If, for example, in our case, the wild type cells are not able to have different adhesion strengths depending on the cell type. I believe you could handle that with some custom functions, but it's not an inbuilt feature right now. Is that correct, Randy?
I would say you nailed it, yes. And I was about to copy paste a NanoHub app, which demonstrates that's the PC3 types is basically differential adhesion if I'm not mistaken, and I was also scanning future presentation sessions. I thought we had discussed at one point perhaps doing a demo of differential adhesion, but I couldn't find anything specifically. So
Great.
I will copy paste the PC3 types, and you can actually look at that source code. But as John pointed out, it does indeed involve editing the C++ code.
So while I'm thinking about that, I think, and you'll see this demonstrated later in later sessions, this as it is, the model builder GUI is limited at this point in time because it doesn't allow static phenotypes. However, having run both raw C++ myself, and then later as PhysiCell developed edited XML myself, and now using the model builder, I think it'll be, I'm really looking forward to using it for just model development and parameters. It makes it much easier to interact with for me. So you'll see how you can use the model builder to change parameters even after you've already written some custom C++, is what I'm trying to say. Great. That is an error. I'm going to take the model. So the last thing we need to do is with a GUI right now is we need to save. Right now it's limited to saving the file to be named mymodel.xml. Of course, you can always change that if it suits your purposes later. So in our case, I wrote it as if that's what our config file is named. So I'm going to move this off screen. And now to run the model. You should be able to just copy and paste. So here we go. All right, it's running. At least on my machine. Hopefully it'll move on to. Oh, great! So we've got our first full output. You can see that it's growing. I think this is actually a little bit of a bug that it starts with 9. It starts with a false cell right now. But so 10, 14, 21, 27 cells. In my case, it takes maybe about a minute to run. With Zoom running, it might take a little bit longer. I'm not sure. My intention is for it to be certainly no longer than 2 minutes on your machine. So if it is, please let me know so I can revise this for future presentations. Yeah, so this is kind of where we're at, waiting for output like usual. I'm going to go ahead and go to the next slide. Maybe you can hear my computer ramping up. Oh, that's something I didn't prepare. I see your question, Genevieve. So I was assuming that the model builder files are in the same level. Actually, I can just show you. So this is root for me. So I've got my PhysiCell download, and then I've got the model builder. And so then I can go to config and invoke the model builder. And then that lets my model.xml right now, it'll save to wherever you've invoked the model builder. So I just wanted it in the same relationship in the file tree, this directory and this directory. So it looks like the run completed a little bit longer than I had hoped, perhaps, but it's done. So we can look at the output. So I'm going to go back to my slides here. Switch sides. So there's a couple different ways you can look at output. A lot of times when I'm running, I like to check. I mean, I always do this when I'm not talking to people. I'll check in my preview. Every system is going to be different, but I just check to make sure that I ran the right executable, that I ran the right XML, and that it meets my expectations. It's always a good idea for a long simulation. And one way to just even get a little bit of quick take on dynamics, of course, is just to run through the preview. I'm running a little too fast, apparently. And then this will open in, who knows? Oh, good. It looks like I didn't. So then you can open it directly in your favorite browser as well if you want a more detailed take. And that's what I did here. Is this is just a tab. So the next way to that we can do to visualize is to make a GIF. So here's the command. This assumes that you have ImageMagick installed. And I think that this takes maybe 10 to 15 seconds. I've found that in the past, this has been a couple of years maybe, but in the past, when I did this command, it could take quite a while if the file sizes were large. So there could be other ways to handle it that might be a little bit faster if you wanted faster feedback. So here's my example. And then I'll show you what it looks like live. So it will be it'll be different each time. But similar, so in all cases, noting that this is a stochastic model, in all cases, we would generally expect the gray cells to grow the slowest and to stick together and not move. The red cells will grow faster, but otherwise have similar behavior to gray. The yellow cells move and randomly and grow faster than red, which is the red is the hyperplastic, so then the yellow is the cancerous, and then the green cells, you can see them moving to the boundary. So they're chemotaxing to where the oxygen is coming in on the boundary due to our Dirichlet boundary conditions, or constant value boundary conditions. And then if I'd let this go on for maybe another day, the green cells would have just covered everything. If you will, you'd have confluence. So that's, of course, a very important step in modeling. In general, is you got to make sure that you're doing what you intend. Great. Are there any questions at this time? And also, if you need, please use this time to catch up. And if I get to the time of moving on and you haven't caught up, please raise your hand and we'll stop. So I see.
Daniel asked.
Okay.
Yeah, if you see his last question there, Daniel.
Great, thanks. So I see Daniel's question, isn't the random seed set in user params? Why would that not lead to identical outputs? Great question. What that leads to is, and you might be able to, and I didn't know this, but apparently PowerPoint will let you pause these. Well, it was letting me pause them there. Pause at the beginning. What it leads to is, so I've got this one's already split, but I've got red, wild type, aggressive cancer. I mean, so you can kind of roughly see this pattern, right? Now I go to here, and I have the same pattern. So that's what that leads to. Is in this case, all it's going to lead to is that right, but I'm missing the most important part. Really, it's threading. That's the answer is threading. So I had 6 threads running on my computer. And if I'd ran it single threaded, then yes, it would be the same simulation every time.
Yeah, we've written an entire paper on that very topic and just haven't published it yet. Reproducibility.
So yeah, and so because of threading, you end up with different birth processes and different mechanical processes because motility processes in this case, because motility and birth and death are all modeled as stochastic processes. Okay, I see another question. I do have the files in the config folder, but can't find the project.exe. So the project.exe should be, again, I've got this live, why not? I can just show you. It should be one level in the hierarchy above config in the file hierarchy. So here's my XML. And then one level above that is my executable, which is going to be without, it's project, should be project. So I just can't scan fast enough. Project, there it is. So you'll want to be in PhysiCell, the PhysiCell directory to, that's, you can see that here. I'm in PhysiCell, and then that's where my command, in my case, running on a Linux-based system, apple.project, is able to invoke the executable. Yes, you do have to make as well. Absolutely. And I hope I went over that. I apologize I went over that at the top of our session. So if you, that was kind of, we set up our tools and our toolkit first, and then went into with our toolkit open and using the model builder tool, we actually sort of implemented our computational model from our mathematical model. But if you hadn't made it the project before you ran through building the model, then you'd have to make as well. I see Thalia asking a question. Yes. Short answer. It's very very straightforward. And so I think that's, in fact, the model builder comes with, I'm going to bring it back onto screen so I can just show you. Actually, I need to be really careful to make sure I don't want to mess with the XML that I have loaded right now. But you can load all of our sample projects, for example, their XML. And because the C++ in that case is already written in, if you either don't want to, you're not comfortable, you're whatever, if you don't want to get into the C++, you can just load, say, for example, the biorobots example, and then use the model builder, not necessarily as you can build with it. And also you could just explore the model, change the parameters, see what happens.
To clarify, Talia, you do not save it back to the same XML file. The shortcoming with the model builder currently is that you can only save to mymodel.xml. Just to clarify.
And then
improvements are coming.
And then in that case, let's see, I don't remember what biorobots compiles to, but pretending like it compiles to biorobots, then you would do config my model.xml, or you could overwrite the existing XML with your new XML. Okay. I probably have a question slide after this, and I feel like we took questions. And I don't see any raised hands at this point in time. And so hopefully everyone had adequate time. If not, now is your chance to scream and shout. So looks like we do have time to do a little bit of model exploration or model building exploration. So I can demonstrate, this will also demonstrate a couple other things that the model builder can do. So I thought it'd be really interesting to look at I see this question. I happen to see the question. So I do not believe that you'll need to, you shouldn't need to use the model builder. If you went through all the steps, you shouldn't need to use the model builder again. Although I don't know what would have happened to the mymodel.xml when you did make reset. Randy might be able to speak to that behavior, but honestly, I'm not sure what the behavior would be when you do make reset for any existing XML. And Randy, could you perhaps address that?
I'm a little fuzzy on the actual question. Divyaj is saying miss the make. So there is no make reset here. It's just,
so at the beginning of the session, the first thing that we did during this time, before we got into the model builder, was actually we made the project using the make command. And so if you look back at my particular slides, the first command that I give is make reset, and then I have make data cleanup, and then make template, and then make make. And so I'm understanding Dvorak's question to be will you have to go over this process again that we just went through and all these previous slides to set up all the cells, for example? And I would think no, unless something like if you did actually rerun everything that was in that very first like open the toolkit slide. I wasn't sure if make reset would have removed the my model XML.
No, I mean that's actually one benefit of only saving to my model.xml, right? It's going to bypass the copying over new stuff. When you do a make whatever project, it copies over new files into the config directory. But since it doesn't copy over anything called my model, we're safe. So
Oh, great.
now, good, great.
Good news, wonderful. Thanks, and thanks for all the questions and making sure that that we're all staying together on this. So for I've got a couple model variations, and these are also in the XML. It's variation 1 and variation 2. It's not, I don't know, it's mind-blowing, but excuse me, it'll demonstrate a couple different things. In particular, I think it's really interesting to use an initial cell profile at times for this. So in order to do this, we're going to change the shape of the domain. So we're going to make it into a rectangle instead of a square. So xmin and xmax go to -500 and 500 respectively. Everything else we're going to leave the same in terms of meta parameters, but we need to click config /cells.csv, and in this case it'll cause the XML to load a CSV file that we're going to populate with cell positions. Next step, because we're going to load the number of cells directly, we don't want any randomly seeded cells. So we need to have enter in 0 for the number of cells. And then that's all we're going to do within the model builder. So when you hit save my model.xml, it will overwrite your existing XML. You should be able to go back through the slides and reproduce that, or just, I mean, I'm not changing a whole lot here. So just wanted to give you a heads up on that. And there's also the rest US XML that you can use. So now open, either use the CSV that I supplied or open your favorite CSV editor like Excel, for example. And then you'll need to enter this array into the CSV file, and I'll explain that as well. Oh, I've got my cheat sheet right there. Nobody needs to see that. Actually, I apparently left this loaded, but what you'll see when you get started is actually this, all zeros. And so column in the first column of the CSV encodes the X coordinate we're starting with the center of the domain being 0, 0. The second column encodes the Y coordinate. In this case, we're placing everything along the X axis, so they're all 0. This third column is for Z, which we're not using. And then the final column is for the cell type. Notice they aren't string names. So that's, you know, looking only at the model builder itself, it's perhaps relatively, it's not very intuitive. If you followed the same procedure as outlined in the slides, then the wild type will be cell 0, hyperplastic for 1, cancers for 2, and then the aggressive cancer will be cell type 3. I'm cheating a little bit. That's not good. I haven't experienced that on my system. I have experienced some quirks, but not that quirk. Good to know though. For what it's worth, and just to be clear, everyone, Genevieve put into the chat the GUI crashes if you try to open the version saved XML file. For what it's worth, for the rest of this demonstration, you should be able to invoke the existing XML that I gave you. Hopefully that will work at the command line. You'll either have to change its name or change the command. So since I already have my CSV loaded with the correct values, and I've already changed my XML, I'm going to go ahead and execute. And now I can check right away that it worked, and it didn't, probably because I didn't hit save. So I can see right away that I'm not getting the behavior that I want, because I don't want a wild type cell at 0, 0. Now I'm seeing output that is correct. I'm getting 2 cell types, and nice that you get immediate feedback here on what the actual name of the, a human readable name. So I can see that I'm getting what I intended, and I'm using the CSV file. I'd like to point out also, I meant to point this out earlier, this is another way that you can check that you're, in addition to looking at the first output from PhysiCell and a preview, you can also look back at all this kind of metadata that gets printed out. So for example, I can see that aggressive cancer has a mean duration, cycle duration of 720 minutes. And that's true for many of the parameters that are modeled to the GUI. I'm switching over to output, so I'll be able to view my output. Great. Again, I'm seeing a rectangle as I had hoped. Great. That's an interesting error. So this will be done in just a few more seconds. Great. Now I'll make it into a GIF. That error, I'm guessing based on that error that our cells got really close together. This warning, FYI, very small vectors are normalized to a 0 vector. While that's waiting, now you can see what my previous run of mine looked like. Now you see really distinct cell populations. They don't interact with one another. They are not sort of overlapping. So you can see the individual behaviors through the duration of the population behaviors throughout the duration of the simulation. Overall, though, to me, it meets our expectations. Again, this is the slowest growing population, next slowest, faster, and then kind of explosive here at the end. They move around quite a bit. And yeah, so I think largely comparable. And I see comments coming in on the yep. So
Yeah, we have a few questions, I guess.
So thank you, Daniel, for reporting that was what your experience. Tara, I'll pop up the GUI again. So in order to use a CSV file, and some of the sample projects use them. I think the worms in particular uses one. You need to check this. So unchecked, checked. And then if you're still getting that after you know that you've had this checked, of course, it's important that if you didn't resave, if you didn't go to file, save, then, well, that doesn't make any sense, what I was suggesting.
I'm afraid some of these errors we would need to see exactly what's being spewed back at you.
Okay.
So you might report with either screen capture or just copy paste text perhaps into the Slack channel. Troubleshooting. Anyway, Sarah got that fixed with
Okay.
checking the box. Great.
Thanks everyone for helping everybody out. This is a great example of community.
John,
So
I'll just point out you're at the bottom of the hour. Are you still okay on your time?
yes, I thought I had until 5.
Yeah, that's what I mean. So
Okay.
we're at the bottom. Very
Great.
good.
Plenty of time.
Great.
And I can go ahead and run through the last demonstration. I see Cecilia still has this question, an issue saving the CSV file.
I'll try and address that. You just
Okay.
proceed.
Okay, thank you, Cecilia, Cecily, for bumping that to the top of the queue. Great. So in this demonstration, I'm going to build in this final demonstration, kind of just show one of the things that I found was really interesting as I was developing this model and working with this model is just a couple of things that I thought were interesting was how much stochasticity did end up affecting the final cell population. So if you just run this over and over again, you'll know there can be some significant variations. Maybe nothing, in some sense, another way to put that into relief is to turn off stochastic birth and death. Actually, I'm sorry, pardon me, I'm going to turn off only stochastic birth. So in this case, I'm going to just leave everything the same as it was before for a minimum of alteration. So the domain's the same. And what I need to do here is I need to go to wild type, go to the cell cycle, the cycle. And then if I just click fixed, now I've effectively turned off stochastic birth, and repeat that for every cell type. So hypoplastic, cancerous, and aggressive cancer. Again, file saved, and it should be ready to run again. All right, so I'm loading the CSV file, and I can, I believe I can check to, looks like we don't have, I don't know that we may not have that output to, not everything is output to terminal. It looks like stochastic birth and death being turned on or off is not one of those things. And in the meantime, while this is running, it'll take, looks like about another minute for mine to run. Are there any questions? Any additional questions? Robin, yes, the way that I invoked this, the model builder will make it run, or we'll make it, I didn't go up far enough. We'll make it save to PhysiCell config. And I'm happy to post that command again. This assumes that you're in the PhysiCell/config directory when you enter this command. Yes, Robin, and we noticed the same thing when we were doing all the work preparing for this, and so that's ultimately why we're suggesting this command. So Robin, again, she said she's having to copy and paste the XML from where presumably I'm guessing PhysiCell model builder, the PhysiCell model builder directory into the PhysiCell/config directory, and this is a solution around that. All right, I'm going to do make GIF again. So this will, that's please follow along at home. But I have already prepared this. Come on. just show this off full screen. And you can really see the differences here. Without the stochastic birth and death, I'm sorry, the stochastic birth, the population remains somewhat limited in the green cells, and then you can really you can see these populations expanding in a very deterministic fashion, as if you've taken a cell line and got all the cell cycles at sync, so that to do an experiment on cell cycle, for example. I mistakenly didn't insert a questions statement pause there, but this is the end of our this is more or less the end of the session, other than a wrap up. I'm going to go ahead and do a review of the session, and then we'll have open questions. So in this session, session 5, where we go over the intermediate modeling workflow in PhysiCell, our goals were to review the intermediate modeling workflow itself. And then basically we did that directly by implementing it. So this bullet point here, more or less, is very similar to the intermediate modeling workflow. So so our implementation of the workflow was, we state the problem. In this case, it's our steps in carcinogenesis model, where the steps are sampled all at the same time, as opposed to watching the evolution of carcinogenesis. So you'd have to think about getting it from either multiple cell lines or multiple samples. Then we planned the model and set up each cell's proliferation rates, the microenvironment, et cetera. So we made the mathematical model, and then using the command line PhysiCell, using the make commands in PhysiCell, we made the template project, and then use the model builder to add on the depth to that project. The template project by itself has one cell type, for example, and is limited. Not much is going on with that. In fact, it's intended to be able to build straight off of. And so that's what we did. So we set up 4 different cell types. So we set up our domain and microenvironment to meet our specifications in the mathematical and conceptual model, and then set up our full 4 cell types. Then we ran and viewed the results, and then even iterated a little bit on it and developed the model further, so that we could see the cell populations spread apart instead of interacting with one another mechanically. We split them apart so we could view to really enable us to view qualitatively just how different and similar they are or aren't, and whether or not that's going to meet our modeling goals. And how then you could further iterate on that if they didn't meet your goals, to alter our mathematical model, or even the conceptual model if needed. So that's exploring the model. Looking forward, the full modeling workflow will be discussed tomorrow. In the morning, Paul's going to talk about what the custom functions are, and that's really important for having a phenotype. For example, a birth or death rate that responds to the environment. So you could think about, we could have thought about, in this case, that maybe as oxygen levels declined, as oxygen levels decline, maybe birth slows down, for example, or death increases. And so these phenotype functions, the whole suite of them, will allow that kind of response to the environment, among many other different things that can happen. And then there'll be a demonstration of the full modeling workflow, I think in the afternoon, my time. Who knows what time it's going to be for you. So in the middle part of the day. Great, and so that's it. Of course, these are folks that have supported. First, thanks of all my lab mates and Paul and Randy. And then, of course, thanks to all the people who are supporting our research. And in particular, the training materials were supported by this administrator supplement. And that's the end of the slideshow. So I'm going to stop the screen. I'm going to stop my screen share and just allow open questions at this time. And I'm happy to reshare as appropriate.
I have a question. Oh, sorry. I think
No, go ahead, Mukti.
Oh, thanks. I have a question that's unrelated to the example per se, but more about PhysiCell. So I've used CompuCell a lot, and that's based on the cellular pods model, right?
Yes.
So then what is the underlying math for PhysiCell?
Sure. The underlying math, I'm happy to actually have these links pulled up so I can share them in the chat, but it's a force-based model. So just like in any kind of like statics or dynamics problem from everyone's favorite classical physics or mechanical engineering course or wherever you may have experienced it. We sum across the underlying model, sums across all the forces that are present on a cell, all the forces that get modeled in a particular instance of the model, and then uses that force to update the velocity of the cell. And then the velocity is used to update positions. Whereas sounds like you're familiar with the cellular POTS model, but just so that it's stated, the cellular POTS model is based on an isinglass spin lattice model. And so it's a statistical physics model. So that, and perhaps maybe being one of the critical differences is that in a physics-based model like this, our modeling units are really, they're physical. So time and dimension, whereas in a POTS model, those all kind of get scaled out and it just ends up being different. Certainly neither's better or worse, it's just different.
Gotcha. Thank you so much.
Of course.
There was one up above. Let's see, where did it go? Stochastic. Andy asked, how does a stochastic birth work? Does it assume some cell type value than 0 and less than duration at start of simulation? If Andy wants to clarify, it's fine. Chime in.
Yeah, I think I might just have to go back to the session 4 and reread or re-listen to that. But I guess I was a little confused about the phase for the birth versus the death and how they could be different times and things like that.
Okay, so how you're perhaps are you expecting a link between the birth and death processes?
Yeah, I guess just the phase length of the birth itself. What does that exactly mean?
Good question. Phase length of so in this case, actually,
Is it like an input value, like
so instead of, so you said birth and that kind of threw me for a second. So the phase length of a cell division, so proliferation. So we're thinking about a cell that goes through all the classic phases of mitosis and or cell proliferation. In the model that I just demonstrated, it's a live cycle model, and it's by far the simplest. So there is no G0 phase. It's not modeled at that level of detail. The duration is whatever we set it to, and it's either stochastic or not. But
I guess what I'm asking, so if the cycle length is like 1,000 seconds,
yeah,
is the stochastic model like assuming some just time value, like it starts some cells at 500 and some at 0, and that's why you get the random kind of division happening? Whereas if it's discrete, do all of them kind of start their cycle counter at 0 and then they all?
I see. Yeah, and I'll do my best to answer that question. And I think going to the source material is where you're going to get the most in-depth answer. And I think Paul would have a really good answer right off the top of his head. Maybe somebody else on the call does too. Feel free to chime in. So no, it's not as if, so in the case of when I set it to deterministic cell death, cell birth, yes, it was exactly like that. The underlying algorithm counts how many or counts how many time steps it's gone through, and then invokes a function to begin the cell division process. So that's why it was you just saw the cell population expanding in steps. So if you have one, it will expand exponentially at the rate that we set it. In the case of doing stochastic birth, instead of it happening at one given time, really it's modeled as a probability. So the probability increases and increases and increases such, and I believe it's modeled as a Poisson process. So the problem, in essence, what you're seeing is the closer and closer you get to the expected cell cycle, the phase duration, the more and more likely it is to happen, until at some point it just becomes deterministic. And so that's one way. So you can use that. And I think maybe Paul talked about that in his slides. I know in an earlier version he had, I think he said like death or something to 9E9. And so then that just makes it so the transition happens immediately because the probability is 1.
Right. Okay, that makes a lot of sense. Thank you.
Yeah, no problem. Great question. I saw another question. I see that Cicely and Randy are still working. Yes, we'll have the, and then Robin, I saw that question. I don't know if your question has been addressed. Oh, good. Thank you, Randy. Then I see a question. This is James O'Neill, a more complicated follow-up question. How does flux balance analysis fit into modeling these forces via PhysiCell? Well, in some sense, I feel like the best answer that I can give you is that it really depends on the modeler and the goals. It fits in however you need it to. And I'm not trying to dodge that question, but I'm interpreting your question to mean you want to know conceptually, is there, how do I integrate the observed behavior that the simulated behavior of a flux balance analysis output into changing the phenotype or behavior of a PhysiCell agent. And I'm not, in truth, that would really be dictated by the model and what your goals were. If you're asking the question that's more algorithmic or more sort of programming based, I can address that in a different fashion.
I think I meditated on it while you were answering other questions, and I think I answered it myself. Just how certain cells evolve in their environments will require different nutrients and higher reaction rates and more motility depending on what they've been pressured to do. And yeah, thanks. I think you answered it well too. So thank you.
Great, thanks. And then as an example of what you could do in PhysiCell without using an additional modeling paradigm, such as FBA, you could kind of like get oxygen from the environment. And this will all be, these kinds of things will be demonstrated tomorrow. You could read the value of oxygen in the environment, and then as it declines, maybe you're tracking, maybe you just even use it directly and say that when my oxygen is below a certain level, well, my speed goes to 0. But there are much more sophisticated ways, such as flux balance analysis, to really take that into account. And thank you for the question and the response.
Thank you.
Alex Weber, does PhysiCell use any sort of calculation cutoff, meaning only cells within some short range are just ignored for calculation purposes? So I believe I'm going to rephrase the question. And so what I'm hearing here is, does PhysiCell have an interaction range for mechanics? Otherwise, you have what, like an n, I don't remember, I'm going to totally mess this up, but like an n squared problem, and you have to do interaction testing with every single agent, and then that'll just be computationally very expensive. And so yes, there is a cutoff range for mechanical interaction testing. I think it's set as a default to be 30 microns, and that corresponds roughly to the default PhysiCell voxel size of 20 microns, and sort of basically you might have to, depending on what your goals are, and kind of ultimately, if you're, I believe I answered your question if you're answering generally, and then for specifics as to what you might want to set that to be, you'd have to get into the details of the problem. So does that address your question, Alex?
Yes, thank you. I was wondering about computational savings.
Great, thanks. So then it gets reduced to no longer being an n order n squared problem. All right. Genevieve asks, so if you wanted to input a specific topology, would you use the coordinates in cells.csv, or would it be better to build a mesh? So I think that I was thinking about that too, and as I was going, honestly, I think it's going to depend on what you're trying to do. In my case, as I was building this model, very simple, and now like I'm thinking about it as a modeler, like I want to play around with some ideas, with some cell types. I don't really know what I'm going to get. I want to run really quickly, like an ideal simulation time is 30 seconds. I think that using a CSV file so that you know where the cells are going to be and you can really set them up how you want is really actually incredibly useful. I was really surprised at how different the simulations were when I seeded them randomly versus when I gave them values. I certainly got different insights and probably better ones with a second setup. And then in terms of, you can absolutely use a CSV file to set up your whole initial cell condition, and you can use other rules too. When you're using the C++, a lot of times I use something that makes a sphere, a circle, a filled circle, and then that I just write into the C++. Great, I see you nodding. Thank you. All right. I'm continuing to go down. It looks like questions have been addressed or are being addressed. So if there's anything, I see 13 messages that I haven't gotten to. If there's anything that, let's see, I see Daniel asking on proliferation rates times modeled stochastically. Is the probability of proliferating in a given time step just R delta T, where R is the proliferation rate? What a great question.
If you go down a little.
Okay, that sounds really, really right. And actually, I meant to put this in the so I can put some of these resources into the Slack. I know we're approaching time, so that they're there. Maybe I'll put some just like the core PhysiCell references into the Slack channel. And then also Paul in particular has a blog post about stochastic processes and it fits in really nicely with how the stochastic, unsurprisingly, it fits in very nicely with how stochastic processes are used and modeled in PhysiCell. And it's a great question. So yeah, I think all right, I'm going to stop talking about it. It sounds like it's addressed. I see Tarunda asking, is it possible to plot the oxygen gradient diffusion? Actually, there are so many questions. Is there anything? Oh, good. I got down to the bottom. Okay, great. I'm down to the bottom. Is it possible to plot the oxygen gradient / diffusion in background in this problem? Yes. And I thought about doing that in this case, but what I wanted to in this particular case, I wanted to stay focused on the intermediate modeling workflow. You saw earlier today Paul doing exactly that. And then he demonstrated just one example of how you could do that. So if your question is, can you do that using the model builder? The answer is no. I think that could be a really interesting function to have in the model builder or in a sort of a whole suite of tools.
I'll chime in very quickly, John. So
Yes.
There's another beta tool in the beta subdirectory called plot data. It's another Python script. So if you run from the root directory of your PhysiCell project and you have everything in your output directory, if you run beta / plot _data.py, it will attempt to do that very thing, plot cells on top of substrates. However, there's more to the story, which we can discuss another time.
I tried to enter that command in there. I don't know if I got it correctly though, Randy.
No.
And then you saw another way that if you have a model on NanoHub or just wanted to explore some of the models over on NanoHub, you saw that can be done there. And then I think everybody has their own scripts, and I imagine in the long run there'll be some as we're doing this exploration of how to best visualize maybe some of the best stuff. Plus Randy will just come up with something awesome anyway. So, but I have some stuff, everybody might have some of their own stuff. So please ask around, and it's very doable and very important. Great. All right. Thank you, Randy. And happy to keep taking questions. And of course, please post to Slack as well. Any questions that you don't want to address right now or that come up later.
Thanks very much, John. That was terrific. And everybody gets 5 minutes back of their life, I guess.
Sounds
So
good.
I'm not following. Oh, here it is, I guess, in front of me. So what is next? Got a 15-minute break. And then at 1:15, there's a brainstorming. And I'm guessing Paul will be back by then to brainstorm on hackathon projects. So see everybody back around 1:15.
Thanks, everyone.
Yeah, I just want to remind that one is in the other Zoom room.
Thank you for coming.